Well, hello, my name is Enrique Ocio from the University Hospital Marqués de Valdecilla in Santander in Spain. And in this presentation, we will speak about IMiDs in multiple myeloma. So these are my disclosures for this talk. And if we go to the treatment of myeloma, we know that in the last century, in this century, we have had 11 novel agents approved for the treatment of this disease. And among these agents, we have some of them that are thalidomide, lenalidomide, and pomalidomide that belong to this family of agents, IMiDs, that we are speaking today. And in this presentation, I want to first focus a bit on the history of these drugs, then how they work, and then what are the differences between them, and if we have any novel agent coming, because as you can see here in this slide, since 2013, 7 years ago, we don't have any new drug belonging to this family of agents approved for multiple myeloma. Everything started for IMiDs with thalidomide in the mid-50s, 1950s, in which thalidomide was used in pregnant women to prevent morning sickness. And we know that it gave rise to the greatest medical disaster in the modern era, when it was called like that because of the presence of phocomelia and amelia. But after a lot of, and in fact, this drug was proscribed and it was not used very frequently. But well, it was used sometimes for the treatment of the lepra. And then in the end, based on the anti-angiogenic properties of thalidomide, it was used or experimented in multiple myeloma. And this brought us to the approval of thalidomide in relapsed refractory myeloma, and later on in combination with dexamethasone for also this setting for relapsed myeloma. And this was one of the, in fact, it was the first novel drug that demonstrated to have single agent activity in more than 3 decades after alkylators or steroids. After thalidomide, then we had other novel IMiDs that were derivatives of thalidomide, and here we had lenalidomide, and of course we had now pomalidomide that we are very used to using them in our myeloma patients. And we know that these drugs are different because these novel IMiDs, apart from thalidomide, they have a more immunomodulatory effect. They have a different toxicity profile. We will speak about that later on in the presentation. And they have, I wouldn't say no teratogenicity, but they have less teratogenicity. So now let's move to the second part, and is what is the mechanism of action of IMiDs? What do they do? Because if we speak about proteasome inhibitors, or if we speak about the acetylase inhibitors or monoclonal antibodies, we know what they are doing, because they are attacking the proteasome, the acetylases, the antigens, depending on the different monoclonal antibodies. But what about IMiDs? Because we don't really know how IMiDs work, or what do they bind. Or in fact, what I will change, we don't know really, we didn't know what it did, what it is doing. And this was until 2010, in which a Japanese group from Ito and colleagues, they demonstrated that the primary target of thalidomide was cereblon. And then when the thalidomide bind to cereblon, that it induced their effects, anti-angiogenic effects, for instance, but mainly they induced the teratogenicity. And so cereblon was identified as the thalidomide binding protein. And in fact, at that point, it was a bit later, but it was thought that maybe we have to change the name of IMiDs as to call them cereblon binding drugs or cereblon binding agents, because that was their primary binding or substrate for binding. And what is cereblon? Well, cereblon is a protein, a molecule that forms together with these two ones, DDB1 or COL4A, they form a E3 ubiquitin ligase complex. So cereblon is a protein that ubiquitinates proteins to make them susceptible for degradation in the proteasome. And here we have cereblon, DDB1, the E3 ubiquitin ligase complex, and we can see how thalidomide, lenalidomide, pomalidomide, and now the novel IMiDs that we will speak about them later, they bind to cereblon, and then what are they doing with cereblon? Are they blocking cereblon? No. they are not blocking cereblon or this E3 ubiquitinase ligase activity. What they are doing is they are changing the activity and they are changing the substrate specificity. So without cereblon, without IMiDs, they have some substrate and they ubiquitinate different substrates. But when they bind to thalidomide, lenalidomide, pomalidomide, to IMiDs, then cereblon changes and binds and ubiquitinates other alternative proteins. And what are these proteins that they bind or they ubiquitinate when IMiDs are bound? The main ones are Icaros and Aiolos. And here there are a lot of biological experiments, but what I want you to focus is that when we have IMiDs here, then we can see with lenalidomide that there is a decrease in Icaros, decrease in Aiolos when we increase the concentration of lenalidomide. Why? Because then when len binds to cereblon, then cereblon ubiquitinates Icaros and Aiolos, and they are degraded by the proteasome. And what is next? So we know that Icaros and Aiolos are transcription factors of different proteins. In concrete, specifically, there are transcription factors for IRF4 and MYC that are really important in myeloma pathogenesis. So if we have low Icaros and Aiolos, then we don't have transcription of IRF4 and MYC, and there's a decrease in the levels of both of them, IRF4 or MYC, both there have a decreased levels. So this is the general schema of activity of IMiDs. When they bind to cereblon, E3 ubiquitinase ligase, then Icaros and Aiolos are susceptible for ubiquitination, they become degraded by the proteasome, and then there is a decrease in the IRF4, MYC, or an increase in IL-2. And then we have all these downstream effects that we know that are immunomodulation, tumoricidal, and also an effect on the microenvironment and in different parts, angiogenesis and different parts of the microenvironment. So I think this is a bit how IMiDs are really working in multiple myeloma and in other diseases, really. In fact, cereblon is very important because it's very important and is required. Cereblon is required for IMiDs activity. We can see how if we in this cell line in vitro, in which they were developed as resistant to IMiDs through continuous exposure to lenalidomide, then we see that these cell lines resistant to lenalidomide, they do not have cereblon, very low levels, by PCR or by Western blot. And we in our lab, we have developed a murine model, an in vivo model of resistance to IMiDs, to lenalidomide and pomalidomide. And if you can see here, these cells resistance to pomalidomide, they have very low levels of cereblon. This is very clear here in immunohistochemistry. And there is no degradation of Aiolos or Icaros. So cereblon is key in the activity of IMiDs. So we can see how all IMiDs really bind to cereblon. And this is the mean of activity of IMiDs. Now the question is, so if all of them bind to cereblon, are all IMiDs the same, or are they different? Because they are all binding to the same target. And we know that they are really different. And we can see, as we have seen something of that before at the beginning of the presentation, that if we analyze thalidomide, len, and pomalidomide through three different cereblon binding proteins, they have different immunomodulatory properties, very few or low for thalidomide, higher for len and pomalidomide. The anti-angiogenic effect is more potent for thalidomide as compared to the other ones. The tumoricidal activity is higher for len and particularly for pomalidomide. And we know also in the clinical point of view that the toxicity is completely different for thal, len, or pomalidomide. Do we have any other information saying that they are different? And in fact, yes. If we go back to our model, in vivo model of myeloma resistance to IMiDs, we developed, as I said before, these cells in vivo that were in vivo resistance to lenalidomide and pomalidomide. And in this case, these are cells or mice in which the tumor growth of mice with cells resistance to lenalidomide. And we analyze what happened if we change here to pomalidomide in these cells. And we can see that if we change to pomalidomide, there is the tumor growth is still controlled with pomalidomide. So they do not have cross resistance. And the other way around, if we have these mice with tumors resistance to pomalidomide. What happens if we change to lenalidomide at this point? And what happened is, again, with lenalidomide, we are able to control pomalidomide resistance. So there is no cross-resistance, and this is important because despite having the same binding protein, cereblon, they are different in the properties, in the mechanism of resistance. So there is something else going on in IMiDs. And now let's go to the final part of the presentation, which is, okay, these are the story of IMiDs, this is how they work, they are different. But since 2013, we don't have any novel IMiD in the arena of multiple myeloma. So are there really any other IMiDs or CELMoDs, how we're calling them now, under investigation? And of course, the answer is yes. And if we go back to this slide, we presented a ubiquitinase ligase complex. We have all this TAL and POM. And then here, we have the novel CELMoDs. And we have CC122, that was the first to be developed, but it was stopped. And then it was replaced by CC220 iberdomide. We will speak about iberdomide now in the next minutes. And finally, we have the CC92480, which is another CELMoD with more potent cereblon binding properties, I would say. Let's start with CC220, which is the one in which we have more data, more clinical data, really, for this drug. This is called iberdomide, which is interesting because Iberia, you know, Iberia is Spain. So I don't know if it's called because of Spain, but it's iberdomide, CC220. And the first thing we have in preclinically is that it's much more potent when degrading Icaros and Aiolos. So it's very potent in the activity with cereblon, and also the activity in the apoptosis and in reducing the cell survival in these cells as compared to lenalidomide and pomalidomide. So this took to the clinical phase 1/2 clinical trial with iberdomide in relapsed refractory multiple myeloma patients. So we can see how 66 patients were included with iberdomide plus dexamethasone. And what is important here is that most of these patients, 86% of them, were refractory to any IMiD, either lenalidomide or pomalidomide or both, which is an interesting situation. What is the safety profile of CC220? Here you can see how the safety profile is quite similar to that of lenalidomide and pomalidomide. We have the anemia, the hematological toxicity. We have also the infections that were also, due to this hematological toxicity, similar to that with lenalidomide and pomalidomide. We have some diarrhea, some fatigue. So the same as we see with lenalidomide and pomalidomide. And now, what about efficacy with iberdomide? What response rate would you expect in this type of patients that were refractory to IMiDs? Well, what we have is 30% responses, approximately, in these refractory patients, and this was maintained in IMiD refractory or in DARA plus pomalidomide refractory patients. Approximately 30% of patients responded to iberdomide. And if we see some of these responses were quite durable, you can see how several of these PRs in green were still ongoing at the time of presentation. So we have durable, good responses, and that some of them were really durable. For me, there is a question here, and we are just almost finishing the presentation, but we have a question, is where would we position iberdomide in the treatment of myeloma? We know that pomalidomide is positioned always after lenalidomide. What about iberdomide? I don't know, but probably, I think I know that probably iberdomide would not be positioned after len or pom, although it can work after lenalidomide or pomalidomide, but probably iberdomide would be independent of them. Even iberdomide would be used even at first line. But we also have another CELMoD, and probably iberdomide would be replaced by this CC92480 that is also very potent and rapid degradation of Aiolos, and also this activity is present in len refractory cell lines. There are two ongoing trials with CC92480, one with dexamethasone, and the other one that would start in combination with daratumumab, Proteasome, or carfilzomib. So this would be the next step in the IMiDs or CELMoDs development. And with that, I finish saying that, we know that IMiDs are now an essential part of many of the current backbones we have in multiple myeloma. They act by binding to the cereblon E3 ubiquitin ligase complex. So they are called probably CELMoDs now. And they also have the degradation through this binding. They have the degradation of ICARs and IOLOS, and then a decrease, a downregulation of IRF4 and MYC. Despite having the same target, they have differential efficacy and biological properties. And we have now novel CELMoDs coming, CC2200 iberdomide, or probably that would be replaced by CC92480, that would be the future. And with that, I finish only acknowledging my team in Santander, in Valdecilla, from the biological and also the clinical part. Of course, my former colleagues in Salamanca, together with Jesus and Miguel, and I thank you for your attention.
Dear colleagues, ladies and gentlemen, I'm happy to present at this meeting now the data on targeting SLAMF7 in the treatment of relapsed and refractory multiple myeloma. My name is Katia Weisel, and I'm talking here from the University Medical Center in Hamburg, Germany. So SLAMF7, and this is an abbreviation coming from the signaling lymphocyte activation molecule, is a target which is highly attractive for myeloma, as it is expressed nearly selectively on plasma cells and myeloma cells, but also in a kind of different role in CD56 positive NK and T cells, as well as in monocytes and macrophages. SLAMF7 is a self-ligand, and in its role, as you see here, in the NK cell activation, it preferentially recruits E2 over the SLAM-associated protein. E2 stimulates calcium mobilization and ERK activation. SLAMF7 signals also through association with MAC1 in macrophages. The first and so far only approved drug targeting SLAMF7 is Elotuzumab. This is a monoclonal antibody, and it was the first antibody approved in the treatment of multiple myeloma. Elotuzumab is directed against SLAMF7, which is expressed on the myeloma cell and NK cells and macrophages, as it is shown again here. The mode of action of Elotuzumab is quite distinct and also special. It has kind of a dual approach, activating NK cells and flagging myeloma cells. As you can see here, the myeloma cell is tagged for recognition by the antibody, and this mediates the cell death via antibody-dependent cytotoxicity. In parallel, there occurs an NK cell activation through CD16, the Fc gamma receptor, and NK cell activation through SLAMF7 when the antibody directs again here the receptor directly via the E2 pathway, as we just heard. And then it comes also by activation of macrophages to an antibody-dependent phagocytosis. So in total, Elotuzumab enables a selective killing of multiple myeloma cells via multiple mechanisms of action. The trial introducing Elotuzumab and led to the approval of this monoclonal anti-SLAMF7 antibody is now published several years ago. It was the ELOQUENT-2 trial. However, it's still very important to mention that here. The ELOQUENT-2 trial tested in a randomized manner Elotuzumab in combination with lenalidomide dexamethasone compared lenalidomide dexamethasone alone in a phase 3 multi-center study conducted in patients with relapsed myeloma and 1 to 3 prior lines of treatment. The endpoint of this trial was progression-free survival, and we have now a very long follow-up on this trial with the published data in a full publication of the 4-year follow-up. Here show you again the extended follow-up analysis regarding the primary endpoint regarding progression-free survival. As you see here, the addition of Elotuzumab to the backbone regimen of lenalidomide and dexamethasone led to a significant improvement in progression-free survival with a hazard ratio of 0.71 compared to the standard lenalidomide dexamethasone backbone regimen. And this advantage was stable over time and again was proven in the 4-year update of this trial. Even as important, there's the analysis of the overall survival. Again here, the addition of elotuzumab to lenalidomide and dexamethasone led to an overall survival benefit, with a median overall survival of 48.3 versus 39.6 months in the standard arm and a hazard ratio of 0.78. Again, this benefit was stable over time. As we know, nowadays, elotuzumab is a very well-tolerated monoclonal antibody. And here you see the updated adverse event analysis of the ELOQUENT 2 trial after a 5-year follow-up. We see here, although it's a busy table, that the addition of the third drug of the monoclonal antibody did not lead to significant more toxicity, especially not in hematologic toxicity or infections. And this was similar between the treatment arms in all analyzed adverse events. Overall, this was nearly the first trial showing that a triplet regimen does not add toxicity compared to the backbone doublet. The currently in the daily clinical life more relevant trial is the ELOQUENT 3 trial. Here, elotuzumab was combined again with an immunomodulatory drug, but here with pomalidomide in combination with dexamethasone. Pomalidomide is the third immunomodulatory drug approved for treatment of myeloma and is the currently approved strongest immunomodulator. In the ELOQUENT 3 trial, the elotuzumab, pomalidomide, dexamethasone regimen was randomized compared to pomalidomide and dexamethasone alone. 105 patients contributed to this randomized comparison. All patients had relapsed and refractory multiple myeloma with equal or more than 2 prior lines of therapy, were refractory to the last therapy, refractory to lenalidomide and a proteasome inhibitor. So in total, 70% of the patients were double refractory. Primary endpoint was a progression-free survival. Here, it's also important to mention that elotuzumab was introduced in a new dose schedule with a 20 milligram per kilogram dose after from cycle 3 on, and then an interval of every 4 weeks, which is more convenient for the individual patients. The data were published 2018 in the New England Journal of Medicine and showed that the addition of elotuzumab to pomalidomide and dexamethasone led to a more than doubling of the progression-free survival rate with 10.3 months for the Elotuzumab arm compared to 4.7 months for pomalidomide dexamethasone with a hazard ratio of 0.54. This resulted in a 46% reduction in the risk of progression or death with Elotuzumab pom dex. The increase in progression-free survival was a result of a higher overall response rate seen in the Elotuzumab pom dex arm, doubling the overall response rate and also leading to more deep responses as VGPRs or CRs. This is even more to mention as Elotuzumab as a single agent does not have anti-myeloma activity and so needs the synergism with the backbone added regimen. Further important results of the ELOQUENT-3 trial showed that patients with high-risk disease defined by the lesion 17p, T414 or T1416 or with an LDH more or equal than 300 units per liter showed the same significant benefit in progression-free survival here with 7.7 months in the Elotuzumab arm compared to 3.6 months in the pomalidomide dexamethasone arm and a hazard ratio of 0.55. For the daily clinical practice, it's also very important that the benefit was equal towards Elotuzumab pom dex in patients with equal or more than 4 prior lines of therapy, showing the same PFS than the total population. The Elotuzumab pom dex trial showed also a significant overall survival benefit with a hazard ratio of 0.62 in the first analysis. Looking here to the adverse events, the ELOQUENT-3 trial showed again that the addition of Elotuzumab did not add significant toxicity to the backbone regimen. Here, the rates of neutropenia and infections were lower than in the standard pomalidomide and dexamethasone arm, although patients were longer on treatment in the Elotuzumab pom dex population. Based on these data, the triplet was approved by the European Medical Agency last year. Looking now into the future, it is clear that SLEMF7 is still an attractive target for treatment developed for treating multiple myeloma disease. However, so far, there's not a breakthrough we currently look on in all the attempts targeting again SLEMF7 with novel techniques. There are currently investigations on cytotoxic T cells, CAR T cell constructs, and antibody-drug conjugates. There was a trial testing an antibody-drug conjugate, ABBV-388, in relapsed and refractory multiple myeloma. However, this development unfortunately was stopped due to the lack of efficacy. Regarding CAR T cells, there are currently trials on the way. We are looking on the first data and waiting for the publications. So far, there's very, very limited data out for CAR T cell constructs with this target. In mice, there were some promising preclinical data captured. So concluding this, we can state that SLEMF7 is a selective and still highly attractive target for developing anti-myeloma treatment strategies. Elotuzumab is so far the only approved drug targeting SLEMF7 as a monoclonal antibody. The combination treatment of Elotuzumab with lenalidomide and dexamethasone was established early is approved, safe, and effective from first relapse on. Combination treatment of elotuzumab, pomalidomide, and dexamethasone is an effective treatment option in higher relapses and leads to sustainable responses with a significant benefit in progression-free and overall survival compared to pomalidomide and dexamethasone alone. The second monoclonal antibody approved for treatment of multiple myeloma, the anti-CD38 antibody daratumumab, which is now consequently entering first-line treatment, is given now to a vast population of myeloma patients. We know that it acts on natural killer cells. However, so far no data exists if previous daratumumab treatment might impact the efficacy of elotuzumab. So far, we are waiting data elucidating this problem. We don't have that in the moment. An antibody drug conjugate, as I mentioned, did not show efficacy in relapse. New immunotherapy constructs targeting SLAMF7 are currently under investigation, including CAR T cells, and first published results have to be awaited. With that, I would like to stop, and I would thank my team here in Hamburg for the view on the Opera House and the beautiful harbor, and I thank you for your kind attention wherever you listen. Thank you.
Hello, everyone, and it's my pleasure to present to you today novel alkylating agents in the treatment of relapsed and refractory multiple myeloma. My name is Dr. Paul Richardson, and I'm the clinical program leader and director of clinical research at the Jerome Lipper Multiple Myeloma Center at Dana-Farber Cancer Institute, Boston, Massachusetts. I want to focus today's discussion on the very interesting and promising novel targeted agent Melflufen, which is actually a very interesting first-in-class anti-cancer peptide drug conjugate that delivers a targeted alkylating payload into tumor. And its full chemical name is melphalan-flufenamide, and obviously that points to the fact that there is a melphalan-based warhead that's part of it, but its actual chemistry is quite complex. So there's the nitrogen-based alkylator moiety, but very critically, the key piece of this molecule is the carrier, the so-called p-fluorophenylalanine, which generates the differences in terms of its properties. Now, in terms of its properties, one of the most important is that it's highly lipophilic, which is in contrast to its great-great-grandparent melphalan. And in that regard, what's particularly important as well to note is that it's primarily metabolized by aminopeptidases. And these aminopeptidases are zinc-based metalloenzymes that catalyze the cleavage of amino acids from the N-terminus of the peptide. And it's essential for basic processes in malignant cells. And very importantly, it's potently overexpressed in myeloma. Now, in terms of the mechanism of action, as I mentioned, melflufen is highly lipophilic and readily diffuses through the cell membrane. Once inside the myeloma cell, melflufen is cleaved by aminopeptidases to release the alkylating payload, and the hydrophilic alkylator payload remains entrapped within the myeloma cell. This in turn leads to the increased diffusion of melflufen into cells driven by a high concentration gradient. So the outside environment is very different to the inside environment of the cell as a result. And within the nucleus, the alkylator payload is able to induce profound DNA damaging effects, resulting in potent myeloma cell death. And I especially want to acknowledge the preclinical contributions of colleagues, including Dr. Zhang and Ray, but importantly, Dr. Dharminder Chauhan from our own center, who has been central to this preclinical evaluation of this drug in myeloma. Now, at the same time, in this same context, Dharminder looked at melflufen in human multiple myeloma xenograft mouse models. And in tumor-bearing mice, he was able to show that there was a profoundly higher inhibition of tumor growth and prolonged survival compared with equimolar doses of melflufen. And this effect was really quite striking. What Dharminder did then further was to look at myeloma cell lines that were actually resistant to other treatments. And what he was able to show was not only is there an associated significant concentration-dependent decrease in vitro cell viability in several cell lines that Dharminder looked at, but most importantly, what he was able to demonstrate was that it was able to overcome resistance to other treatments, especially Melflufen, Proteasome, and dexamethasone. So provocative information preclinically. In terms of the first in-patient study for myeloma patients, that is, I'm very pleased to show you results from our 012M1 study that was presented at ASH last year, and in fact was published in the Lancet Hematology journal in March of this year. This was a phase 1/2 open-label, multi-center international trial. Our key eligibility criteria for patients with relapsed and refractory myeloma who had at least 2 prior lines of therapy, including lenalidomide and bortezomib, and they had to be refractory to their last line of treatment as defined by the classical definition of refractoriness, with progression on treatment or within 60 days of last dose. Now, the phase 1 dose escalation part was in 23 patients and is summarized from 15 to 25 to 40 to 55. 40 milligrams was defined as the MTD. Should be noted that the Melflufen was infused every 3 to 4 weeks, with dexamethasone given weekly. In the phase 2 expansion cohort, where we enrolled 58 patients, we looked at Melflufen plus dexamethasone on a 21 and 28-day cycle and compared this to a single agent cohort, which was just Melflufen alone. And then finally, with the results of that study, we were then able to follow up these patients for a prolonged period of time to evaluate them not only for safety, but also efficacy as reflected by response rate, progression-free, and overall survival. Now, in terms of the dosing schedule, it's important to note that we were able to define that the 28-day cycle proved to be favorable in terms of platelet recovery and neutrophil count. And we're also quickly able to define an MTD of 40 milligrams. And I summarize here the 21-day cycle and 28-day cycle results to illustrate why the 28-day cycle was chosen. Now, what's very important now to share with you are the baseline characteristics of the patients we treated, because then we can not only discuss the efficacy piece, but also I'll spend a little bit more time in a moment on the safety. In terms of the combination cohort, which clearly was superior, and the single agent cohort was just 13 patients strong. We'll come to some of the results from that in a moment. But in the 45 patient cohort, you can see this was very typical for a relapsed refractory population in the current setting, recognizing that this study started before the advent and impact of monoclonal antibodies on a large scale. Nonetheless, actually 8 of the patients were exposed to either daratumumab or elotuzumab. But as you can see, the vast majority, consistent with the protocol eligibility criteria, were refractory to both the PI and an immunomodulatory drug, i.e. double refractory. Most had received a prior transplant, and very typical, the median number of lines of therapy was 4 with a range of 3 to 5. Similar characteristics were seen in the single agent cohort, albeit smaller. And very importantly, we were enriched for high-risk cytogenetics, with 44% of patients having, and those are valuable, having high-risk features. Now, in the context of the patient disposition in detail, I want to summarize them here. As I mentioned, 45 patients were treated in the combination cohort, 13 in the single agent cohort. And in fact, in that single agent cohort, the side effect profile was very consistent with what we'd seen previously, but the efficacy data was something we'll come to in a minute. And that led to the decision to continue only with the combination cohort thereafter. As you can see, the discontinuations were primarily due to progressive disease and or lack of efficacy versus adverse events. In terms of response, if you look at the combination cohort, we were very pleased to see that the overall response rate in this refractory population was 31%, with MR or better seen in 49%. In the single agent cohort, whilst there was an 8% overall response rate and an MR or better of 23%, clearly this was inferior to that seen with the combination. Now, importantly, in the combination cohort, we had VGPRs actually in 5 patients for a total of 11%. Now, in terms of duration of response, this is summarized on this swimmer's plot. And as you can see, the median duration in the combination cohort was actually encouraging at 8.4 months. Now, in terms of alkylator refractoriness, this was an important part of the study. We looked carefully at this. Basically, what we were able to show was that if you looked at all alkylator refractory patients and who were refractory to any prior line, it was 33% of the patients. And last line refractory was actually quite high, of 5 of 11 patients, so 45%. We also looked then at the Melflufen refractory patients and could see there that the refractoriness was high as well as they were to the last line refractory. And I want to clarify this by stating we had a significant number of patients who were alkylator refractory, 24, I beg your pardon, last line refractory 11, Melflufen refractory 9, and last line 3, I beg your pardon. Now the response rate, this is the critical point, is that they were refractory in any line, 33% of patients were responsive, and 45% were responsive if it was last line. And similarly, if they were Melflufen refractory, it was 44% refractory, and last line 67%. Now, numbers here are small, so I think we have to be very careful. But the point is that we saw patients who were previously alkylator refractory, i.e. both Melflufen, cyclophosphamide, and so forth, or purely Melflufen refractory. We saw encouraging signals in terms of response rates, with the strong caveat that obviously this was a small number of patients. Now, in terms of progression-free survival, we were encouraged to see this, that the median progression-free survival for the combination cohort approached 6 months, clearly being lower for the single-agent cohort. What was particularly striking to us was the overall survival. And if we looked at with the median follow-up approaching 28 months, the median overall survival in the combination cohort for this relapsed and refractory population was 21 months, which we thought was quite encouraging. Now, what about the safety? Well, in terms of treatment-emergent adverse events, these are summarized here, and it's a busy slide, but what I want to emphasize to you is that most common grade 3-4 adverse events that were considered Melflufen-related were reversible thrombocytopenia and neutropenia. As you can see, if you look at the non-hematologic treatment adverse events, they were relatively few, and most importantly, if you look at the grade 3 and 4 events, they were equally relatively few. And amongst the important side effect profiles that were seen, as I mentioned, myelosuppression was by far and away the most. But it's important to note that Melflufen is not associated with alopecia and not associated with mucositis. This is a particular advantage to this drug. Now, in terms of adverse events leading to discontinuation, basically this was primarily dominated by thrombocytopenia, neutropenia, and anemia. Now, we did see some early mortality in the study for representing a very sick population. And this occurred exclusively in the context of rapidly progressive disease, where 2 were considered possibly Melflufen-related and were infectious in nature. Now, in terms of the overall summary, we therefore felt from the IS experience that a dose of 40 milligrams once a month combined with dexamethasone was the dose to take forward, and that the safety profile proved manageable generally, particularly in the context of myelosuppression. Now, in terms of ongoing clinical studies, I want to just spend a few minutes on this. We have the Horizon study, which I'll summarize in a moment very quickly. We also have a very important anchor study that's combining Melflufen with daratumumab or bortezomib. And importantly, there is the phase 3 pivotal trial that's ongoing at the moment, comparing Melflufen and dexamethasone to pomalidomide and dexamethasone, which is actually hoping to complete enrollment later this year. And in the Horizon study, we have presented this at a couple of meetings. Both my colleague, Dr. Marie Mateus, and others have presented this data last year, and I want to share this with you. Basically, this is a relapsed and refractory study, single-arm trial. At the present time, we have 154 patients to report on, a median age of 65, median number of prior therapies is 5. We're enriched for high-risk cytogenetics and extramedullary disease, and the true triple-class refractory penta-exposed group is really quite high. At basically over 70% in both categories. Now, this study is primarily designed to look at overall response rate with secondary endpoints including PFS, duration of response, overall survival, and other parameters including safety. Critically, what we've seen in the efficacy summary here to date, recognizing that these analyses are ongoing and remain therefore preliminary, we've seen an overall response rate approaching 30%, which is encouraging in this particularly sick triple-class resistant penta-exposed group. What's also particularly important as well is that we've seen benefit in patients with extramedullary disease, for which this study has been remarkably enriched. Importantly as well, median overall survival is now 12 months, which is great for the overall population, given how, again, brittle and fragile this patient population is. And we're also encouraged that the PFS has been approximately 4 months, recognizing that it's somewhat shorter in the extramedullary disease group, which is very typical of this group. Now, in terms of the safety summary, there has been really nothing unexpected outside of the myelosuppression that we were anticipating from our earlier phase study. But having said that, there were actually surprisingly fewer infections than we were perhaps anticipating. In that regard, certainly the incidence of upper respiratory tract infection has been quite low. And in that same context, we've had around about a 20% infectious rate reported as SAEs that were not neutropenic. And therefore, in this brittle population, where typically 20 to 30% rates of URIs are often seen, this we felt was relatively encouraging. So the overall rate of grade 3-4 infections about 20%, and again, generally manageable. And of the terminal events that occurred during the study interval, these were seen in 5 patients today, 3%. None were related to Melflufen in the opinion of the investigators, and all were in the context of progressive disease. Now, the ANCHOR study is a particularly exciting one, and this incorporates the combination of Melflufen with daratumumab and bortezomib. These, again, are preliminary results, and I especially want to acknowledge the principal investigator of the study, Dr. Enrique Ocio, who presented this at the ASH meeting last December. And what you can see here is that in the daratumumab arm, certainly, a number of patients have been successfully dosed between 30 and 40, and a smaller number have been dosed with bortezomib. The response rates are really quite encouraging at 76% for the daratumumab combination and 67% for the bortezomib combination, which, given the relapsed and refractory nature of the patients, is quite striking. So in conclusion, Melflufen is a first-in-class peptide drug conjugate that represents a novel mechanism of action for patients with relapsed and refractory myeloma who have few remaining effective treatment options. In our first phase 1/2 study, we were able to demonstrate that there was encouraging activity and durable clinical benefit in patients who were heavily pretreated. We feel that the Horizon and Anchor data are encouraging, recognizing they remain preliminary. Particularly, however, we're struck by the activity in patients with relapsed and refractory disease and extramedullary relapse as a dominant feature, which is an area of unmet medical need. I want to emphasize that the Ocean study, which is led by my colleague, Dr. Peter Sonnefeld, is actually almost complete in terms of enrollment, and this data, I think, will be very important. Critically, the safety profile is appearing manageable, with most adverse events being reversible, and indeed constituting thrombocytopenia and neutropenia. But fortunately, the non-hematologic AEs are infrequent. In that same context, the combination strategies going forward, I think, reflect the most exciting and promising new direction, as well as hopefully moving this agent earlier into the treatment paradigm. I want to close by especially acknowledging all the co-investigators on the studies, and in particular, of course, the study teams, but first and foremost, to acknowledge the patients and their families. And finally, I do want to acknowledge the pivotal science from Dr. Joachim Gulbo, from Uppsala, as well as the team at Karolinska, who actually generated this novel drug in the first place, as well as Frederick Lehmann, and of course, all the team with our sponsor, Oncopeptides, based in Stockholm, Sweden. I want to close by thank you very much for your kind attention.
Hello, I'm Dr. Thomas Martin. I'm co-director of the myeloma program at the University of California, San Francisco. And first, I'd like to thank the organizers for inviting me to give this talk this morning. Second, to the viewers, I hope you're all well. I hope you're on the road to recovery, whatever part of your world you're in, and we're hoping that everything turns around and we can get together soon. So my topic today is CD38. The goals of my session, really, I'm going to briefly review the mechanism of action, the resistance of CD38, the most recent clinical data from Candor and Icaria, and then more importantly, I'm going to discuss some of the future therapeutics, and I'll present some of the structures of some of these novel CD38 antibodies, some that are, I think, really interesting and also might have quite a bit of activity even in the relapse and refractory state or in the CD38 refractory state. So I think we've all seen the anti-CD38 mechanism of action slide. CD38 antibodies work mostly probably by ADCC and ADCP. But as you know, daratumumab was selected based on its ability to induce potent CDC. It certainly is the most potent antibody to induce CDC. And in contrast, isatuximab, the most recent approved CD38 antibody, was selected on its ability to induce apoptosis via direct caspase dependent mechanisms. What we do know also is that there's a significant amount of immunomodulation with decrease in the number of T regulatory cells, B regulatory cells, and myeloid-derived suppressor cells. What we don't know and what needs further information is really how important is the ectoenzymatic function of CD38 and what are the effects on NAD as they correlate to myeloma survival and also effects on the local microenvironment in T cells. There was an abstract here in ASH of 2019 by a Japanese group looking at a novel molecule that they called CD78. And they proposed that CD78 was a potent inhibitor of the enzymatic activity of CD38 and that it can induce cytotoxicity. On the right, you see the cell surface and you see CD38, which regulates intracellular NAD levels. And by blocking the CD38, you have elevation at the intercellular NAD levels and growth arrest. And they showed this in vitro. They also showed at the left side of the panel very high NAD levels correlating with the growth arrest. And they also proposed that this could have potent synergy with daratumumab, which has very little enzymatic activity. Now, whether this is going to really be a mechanism of providing a potent anti-myeloma effect in the future is yet to be known. In terms of resistance, there's primary resistance and acquired resistance, and I'll just briefly say that primary resistance is dependent on host factors, on Fc gamma receptor polymorphisms, and on host factors of KIR and HLA genotypes. We showed at UCSF that an inhibitory KIR and HLA genotype was important to response or no response to CD38 antibodies when isatuximab was combined with lenalidomide and dexamethasone. We're also seeing more data now on CD47 expression levels that don't eat me signal and how that's important for resistance to antibody-based therapy. Now, we know that everybody that started on CD38 antibody therapy develops resistance, and it's likely due to downregulation of cell surface of CD38. Many reasons for that. Probably the two most important. One is some of the clones that are present may actually have less CD38, and those are the clones that are going to rise to the top, and those are the ones that are going to become resistant. CDC and ADCC requires a certain number of CD38 molecules on the cell surface with current therapeutics. And then we know that patients do get overexpression of some of the complement inhibitory proteins like CD55 and 59. You're going to hear more about that. We're going to have coming soon more data on single cell sequencing of primary myeloma cells, but also on cells in the microenvironment. And we know that there's already signals that patients who respond have increased M1 macrophages in the local environment, increased central memory T cells, and increased expression of CD28. Many researchers have tried to increase the expression of CD38 on the cell surface, and I still think that that's possibly a viable mechanism to negate the resistance that develops to CD38. But we need more data, and we probably need better agents to cause overexpression of CD38. Now, as you know, we've had many key studies. And what I think for the present and in the future, the key studies are for daratumumab in the front line. Obviously, the key studies, the MAIA, the Alcyone, the Cassiopeia, those studies have formulated a path for daratumumab to be used in the front line. However, in the US, we don't use those regimens as much. And we've been very comfortable with giving patients lenalidomide, bortezomib, and dexamethasone. So therefore, I think the Griffin study that was just recently published in Blood as well as the phase 3 EMN study of DARE RVD versus RVD, will be very important to the path forward of using daratumumab as frontline-based therapy. DARE has also been used together with KRD. We heard a presentation at ASH from the MASTER study, and we also have other DARE KRD studies that are ongoing. And I do think daratumumab will make it to the front line, especially in the US, likely as a quadruplet. In the relapse refractory space, there's so many studies and so many publications that I can't really include them all here. We know it started with Castor and Pollock. The most recent one, Candor, where daratumumab was combined with carfilzomib and dexamethasone. I'll show you data on in a minute. In terms of isatuximab, isatuximab does have some key studies in the front line ongoing. The EMRO study is randomizing patients to isatuximab plus RVD versus RVD in transplant ineligible patients. The German group also has the HD7, which is isatuximab with lenalidomide, bortezomib, and dex versus RVD in the transplant eligible population. And there are also studies with ISA, KRD. In the relapse study, I'll show you a few slides in Icaria, what led to the approval of lenalidomide, approval of isatuximab in relapse and refractory space. I also want to point out the IKEMA study. It's very similar to the Candor study. It's isatuximab with carfilzomib at the 56 milligram per meter squared, dosing together with dexamethasone. It's a randomized phase 3 versus the carfilzomib 56 versus dex, a doublet. And we should actually see some data from that study relatively soon. So here's the schema of the Icaria study. Involved 300 patients. Again, ISA POM-DEX versus POM-DEX. Standard dosing of isatuximab, 10 weekly for 4 weeks, and then every other week until disease progression or unacceptable toxicity. This is a very heavily pretreated patient population with many patients refractory to PIs, IMiDs, and upwards of 3/4 of them being both IMiD and PI refractory. And this showed a very nice progression-free survival of 11.5 months versus 6.5 months in the control arm. And this was significant enough that it led to approval by the FDA. And that's the one regimen that's approved for isatuximab. Now, what about carfilzomib? I think the most recent study presented was the Candor study. It was presented by Dr. Usmani at ASH this year. Again, looking at carfilzomib, dexamethasone, and daratumumab versus carfilzomib and dexamethasone. Here's the study schema. Standard dosing to carfilzomib twice a week for 3 weeks, with the 4th week being rest week, and then a daratumumab weekly for the first 2 cycles, and then every other week. Again, this was a less heavily pretreated population. Only about 1/3 of the patients were refractory to an IMiD or to a proteasome inhibitor. And these patients had an average prior line of therapy of 2, with upwards of 45% of them having just 1 prior line of therapy. Here's the PFS showing the marked advantage of DARA dexamethasone versus KD. And this has led to approval of carfilzomib with daratumumab in the relapse refractory setting. Now, patients did achieve deep responses, and with some patients, upwards of 12% of patients at 12 months achieving MRD negativity. And this is in the relapse setting, so that's a pretty potent agent in regimen. And I think that will be a regimen that we'll use significantly in the future. I think ISA POM-DEX and carfilzomib DARA-DEX are both regimens that will get a lot of use here in the US and abroad. How can we augment the CD38 activity? There's many avenues that people have approached. One way is to enhance the FC region with glycoengineering. Another is to have better selectivity with the binding domain. And another option is to provide some immune activation or a dual targeting mechanism from the antibody, or delivery of a toxin like an ADC or a specific cellular toxin. We do need better combinations. I think there's many things that are currently being tested, like CD38 antibodies and checkpoint blockade, CD30 antibodies and hypomethylating agents. We have a study at UCSF where we're using azacitidine to try to enhance CD38 expression on the cell surface in patients who are resistant to daratumumab. And we'll see if that is, again, a viable pathway to reverse daratumumab resistance. There will be studies with CD38 plus an anti-CD47 antibody, and we're very excited about those, as well as using monoclonal antibodies, including CD38 with ex vivo expanded NK cells to try to give NK cells together with the CD38 to enhance its effect. And you can pre-expose those to an antibody that will bind up CD38 and prevent killing of NK cells by the CD38 antibody. Well, now let's talk about the novel anti-CD38 antibodies. There's a variety that are currently in clinical trials. Only one, TAC-079, that is available for me to present the data and was presented at ASH this past year. But in terms of enhanced binding and activity, there's TAC-079. There's a second generation CD38 from Sanofi, SAR442085. And there's also a hexabody CD38 that I'll show you a slide on in a second. There's a combined immune activation CD38, TAC-573, that's in clinical trials, where it binds the CD38 and has activation of interferon on the FC domain, and I'll show you a picture of that in a second. We also have a few bispecifics for CD38 in clinical trials. I'm not going to say more about them, but we are worried about the possibility of increased toxicity, specifically marrow toxicity, since CD38 is expressed on some bone marrow cells and some stem cells. Now, here's data that Enrique presented at ASH this past year of TAC-079. TAC-079 selectively enhances depletion of CD-positive cells, CD38 positive cells, and in fact, it focuses its activity on the high density targets. So this takes advantage of the fact that myeloma cells have a very high density of CD38 on the cell surface, and therefore has little off-target effects. And in fact, there is less binding to red blood cells for this, and also less depletion of lymphocytes in patients who receive this therapeutic. Here's the study schema. It was a dose escalation standard, a 3+3 design, where patients started with 45 and went up to as high as 1,200 milligrams with the plan to go to 1,800 milligrams. There will then be dose escalation cohorts. In the 34 patients that they presented at ASH, 90% of them were refractory to a prior line of therapy and 80% of them to their last line of therapy, with a fair number of patients refractory to either an immunomodulatory drug or a PI, approximately 90%. Everybody was prior exposed to PIs and IMiDs. Now, the drug actually was very well tolerated. If we look at the incidence of lymphopenia, thrombocytopenia, or infusion-related reactions, there actually was very few grade 3 events. In fact, neutrophil decreased in only 4 patients, two patients of the 34, and anemia only in 1 patient. There were no infusion-related reactions. And this therapeutic was given by subcutaneous injection, and when there were no injection site reactions. If we look at the activity, you could see the lower doses at the bottom of the slide, the higher doses at the top of the slide. When the colors turn green and blue is when they start to see activity. You could see around the 300 milligram dosing cohort, they started to see some activity, with much better activity actually at the 600 milligram dosing. We look at overall response rates in the intent to treat population at 300 milligrams with 12 people treated. Overall response rate was 45%. At 600 milligrams, it was 12%. And there was some patients who were responsive even though they were DARA refractory. If we look at the progression-free survival, You can see the curves here. The early progression-free survival curves, especially at the 600 milligram dose, looks promising. Now, the authors concluded that these results were quite interesting and showed that the 600 milligram dose is effective, and they are going to continue to dose escalate this drug. They have not achieved an MTD, and again, no dose-limiting toxicities were seen. It'll be really interesting to see this drug in combination with other drugs, including pomalidomide and dexamethasone. Now, the next few slides, I'm just going to go briefly over some of the novel therapeutics that are in clinical trial or that we've seen data at ASH. The first is this Hexabody CD38 Gen 3014. This is a anti-CD38 antibody that forms these potent hexamers, and these hexamers actually are much better at fixing complement, and they enhance CDC. So this therapeutic has a much higher degree of CDC, even of daratumumab, our most potent CD38, but also induces other means of toxicity, including ADCC, ADCP. It does do immune modulation. It affects the CD38 enzymatic activity, and also there's some ability to induce apoptosis. So it's a very interesting drug. This is some in vitro curves of the ability to induce CDC in newly diagnosed patients in the panel A, in patients with relapsed or refractory disease in panel B, and kind of the culmination of multiple patients tested in panel C, showing newly diagnosed patients, relapsed refractory patients, and then DARA-treated patients, where it's a little more difficult, obviously, to induce lysis in the daratumumab-treated patients. And it is possible that those patients have more difficulty with CDC, basically because of decreased cell surface CD38. Now here's two other novel molecules from Takeda. You saw the first one, TAC-079. On the top is TAC-573. This again is an anti-CD38 antibody, an IgG4 fused to an attenuated form of the human interferon alpha. And basically this binds to the cell surface, and the interferon moiety binds to the cell surface interferon receptors. It can activate interferon-mediated signal transduction pathways, and this may actually enhance the anti-myeloma effects of the CD-positive cells. It may also have some effect on the local microenvironment. On the bottom is TAC-179, which is another interesting molecule. It's an engineered toxin body. It has an SCFB to CD38. It binds to similar, it overlaps a little bit with daratumumab, and then it is engineered to have a Shiga-like toxin, the alpha subunit. And this is a very potent toxin against myeloma cells. You can see in an animal model, a xenograft model at the bottom, a significant anti-myeloma activity. And this is on the right in the curves. You can see that there's activity against primary myeloma samples from patients with multiple myeloma. Now, both of these are actually listed in clinicaltrials.gov, with TAC-573 having recruitment ongoing, and recruitment has not started yet as far as I know for TAC-169. Now, again, there's other strategies for CD38, including enhanced FC domain of the antibody. That's the Sanofi molecule, and that also is in a clinical trial at the current time in the US, France, and Spain, and we're hoping to see some data in the next 6 to 12 months. And then there are a couple CAR-T trials that are listed on clinicaltrials.gov. One in the US with a CAR-T that specifically, like TAC-079, binds to CD38 in high density, and hopefully will save on some of the toxicity. With that, I'll conclude that targeting CD38 is here to stay. We need to better define resistant mechanisms and ways to really enhance our anti-CD38 activity, especially with hopefully the ability to overcome resistance. There's several next generation CD38 antibodies and therapeutics that are in clinical trial. I think the combinations of IMiDs and PIs obviously have shown promise and will continue to show promise. And I'm looking forward to seeing some of these novel drugs in combinations. It is unclear at the current time, I think, if immune activating strategies like bispecifics or CARs directed against CD38 will actually be successful or too toxic. And we'll just have to see how the data plays out over the next 6, 12, 18 months. Thank you very much for your attention.